Good afternoon, everyone. Thank you for joining us. My name is Claire. I'm a contract librarian on assignment here at NOAA Central Library, and I'll be your host this afternoon. This seminar series is offered through the NOAA Central Library Seminar Program, which provides an educational forum for the presentation of ideas, research updates, and news in support of NOAA's mission. Before we start, I have a few technical tips for attendees. Just so you are aware, you are muted and you're not able to unmute. However, if you have a question, you can type it into the question panel and we'll get to those during the Q&A session at the end of the presentation. If you're having trouble with audio or seeing the slides, you can try logging off and logging back on, and that will solve most issues in GoToWebinar. And as a reminder, this is being recorded. If you would like to revisit this seminar or view previous seminars, you can find them on our YouTube channel, and I can place a link to that in the chat for everyone momentarily. Now I'm going to turn it over to Peg, who's going to introduce our speaker today.
Thank you so much, Claire. Yes, this is Peg Brady from NOAA Fisheries. We're continuing our 2021 NOAA EBM, EBFM seminar series, which we initiated in November of 2017. And our objectives have been really to showcase the excellent projects that are being implemented by NOAA and our partners to advance ecosystem-based management, to increase awareness regarding EBM, and by highlighting initiatives that are being undertaken by our staff across the country, and to enable our fishery staff to highlight their own ecosystem work. NOAA Library, as you just heard, has been our co-host for this series, and the seminars are held the second Wednesday of each month from 3 to 4 p.m. Eastern Time. Each presentation is publicly accessible, recorded, and archived, thanks to the library. And as Claire had just mentioned, all the recordings are available. So if you know of anybody that will miss today's presentation, certainly share that link with your colleagues. And then obviously we have some questions that folks will be generating, and Claire will help us or focus those questions to Jim. And also there's an opportunity to follow up with Jim after the seminar today. And thanks to Jim Thorson, who was a scientist at our Alaska Fisheries Science Center, who's going to speak today, especially on a topic near and dear to my heart, and that is habitat. The title is The Grand Challenge for Habitat Science, Predicting the Impact of Local Human Activities on Regional Fishery Productivity. Jim works very closely with the Alaska Fisheries Science Center folks and the regional office, as well as our other staff that work on EBFM across the country. And I want to thank Jim for taking the time out today to present to our series. Just one note, we do have a speaker lined up for October 13th. That's Avalon Bristow, who will be giving us an update on the Mid-Atlantic Regional Council on the Ocean, and continuing our series about updates from all the ocean planning councils across the country. So again, thanks, Jim, and we'll turn it right on over to you. Thank you.
Yeah, thanks. Can you confirm that you can hear me?
Yes, we can, Jim. Thanks so much for checking.
Great. Yeah, and before I get going, thanks Peg, thanks Claire, and thanks to many others for making this possible. Of course, everything's always a relay, and there's more people to thank than I have time. So I guess I'll start with the obvious. I'm standing here at home in my bedroom. Actually, I imagine many of you are at home. Here's a picture of me with my recent 13-month-old Mira. When we, kind of speaking below the neck for a second here, when we decided to have a second kid, of course, we didn't know that we would be having them in this sort of moment where they, where they get to know our house extremely well. And it's given me a lot of time to think about, how interesting habitat is. Habitat, of course, comes from the, Latin root for home. And it's the home, when we talk about habitat, we're talking about the home for animals and for people, the things in the home that allow them to thrive. And, as our moment is showing, our habitats, our homes, and our well-being are really tied via teleconnections to things that are geographically distant. You know, and as climate change will continue to occur, you know, we really need a way to think about how distant events and distant things, maybe across the world, can end up affecting all of us in our homes. And that's sort of the center of what I think is the Grand Habitat Challenge. How do we predict the impact on our homes and on animal homes from things that are local, but also things that are geographically distant? And how do we help everything thrive in their homes? So let's see now. Here. So you know, a more technical definition is given by Planck in this paper. Habitat is the geographical realization of niche. Niche is another one of these words, you know, it comes from nest. And so, you know, when we talk about habitat, we need to keep in mind that we are talking about the, you know, where animals live, as well as the attributes of where they live that make it a good place to live. And so, you know, as I'll talk later about, there's sort of impacts on habitat directly on the characteristics of a place that make it a good place to live. But then there's also times when, you know, we can directly impact the place they live by trawling or so forth. And those are also habitat impacts. They're not mediated through the qualities of the habitat. They're just directly scooping up all the fish in a given home. And so today it's cut off on the left-hand side, but I'll quickly introduce the Grand Habitat Challenge. I'll talk about how to get moving. You know, how do we think about movement of animals in this? I'll talk about who's hungry. How do we think about predation? And then I'll end with a short slide of where this gets us as an agency. So the Grand Habitat Challenge is, you know, something that I and co-authors put forward. It's published in ICES. And it specifically has this longer title about stake-structured responses, non-local drivers, and mechanistic associations among habitat variables affecting fishery productivity. So my observation starts with the idea that in our science at NOAA, we've been using, by and large, this sort of pressure indicator response framework to habitat studies. So you know, if we have an activity like bottom trawling, we might first look at sort of how that pressure affects an indicator, like, you know, the structure forming invertebrates in rocky habitats or in sandy habitats, you know, benthic infauna. And, you know, so we might do a single study doing experimental trawling with a specified type 1 and type 2 error rate to look at, you know, basically answer the question, is trawling affecting this indicator, like deep sea corals, for instance? And then if we find, yes, that it is significantly affecting deep sea corals, it's not minimal and more than temporary, then we might secondarily do studies looking at how those habitats, how corals affect condition, juvenile density, or food at disturbed versus undisturbed habitats. And so, you know, this sort of stack of work is incredibly important and will always need to be a part of our habitat work. But it really ties us into these specific indicators and ties us into a specific experimental approach of you know, identifying an indicator, a habitat indicator, and then identifying how that affects species of interest. And the problem, of course, is that we're in a world where there's way more human impacts than we're able to do rapidly in this way. So instead of just impacting structure forming invertebrates, you know, fishing affects the distribution, the abundance, and the forage of top predators, and that can directly affect habitat quality. We're also conducting a massive unreplicated and unplanned experiment in what happens if we increase the temperature of the oceans, and that's affecting habitat. And we need to be providing information about a broad range of things like this, like what happens if we deploy a bunch of wind farms in the northwest Atlantic. You know, what if we start rebuilding coastline habitats in the you know, in near Louisiana? These are all studies that are hugely important and need an analytical framework that allows us to look at multiple impacts and do, you know, not just a pressure indicator response framework, but an analysis of trade-offs. How does wind farms not just do affect an indicator, but how does it affect all of the things we care about in management? So as an alternative model to this pressure indicator response framework, I point to what we do in stock assessment. Stock assessment is a very successful field that's evolved hugely over 80 years. It continues to be repurposed for new uses. And in this, we typically, describe stock status relative to targets. Those targets can be discussed in our kind of co-management system, our council process. But we have kind of analytic machinery that we agree upon worldwide. And so that machinery basically involves looking at a change in biomass. And that's a function of current biomass via density dependence. It's a function of environmental responses. And it's a function of external drivers like catch. And we do that within this biomass dynamic model that's been continually extended over the years. Habitat, by contrast, has gone down this different framework of developing species distribution models, or SDMs. And they typically are defined to look at biomass, not a change in biomass, at a series of sites. And I'm sorry, I need to fix the labeling on those Bs. So biomass at different sites, S1, S2, and S3, as a function of local environmental conditions at those different locations. So this SDM has a couple issues. It doesn't represent population dynamics. It doesn't include harvesting. It doesn't include density dependence. And it doesn't really look at changes in biomass, which is typically what we care. We care about how humans will affect the change in productivity, not just a realized utilization distribution. And so, I guess I start by pointing out that researchers have generalized, they've already fused these two things where we can do these sort of demographic models at every location, S1, S2, and S3, including density dependence, environmental responses, and catch as an offset. We know how to do this analytically. We know how to do it accounting for residual spatial correlations, but it hasn't been used in habitat analyses as much as it could be. So, why is it not being used as much? Well, there's, of course, different analytical challenges. One is stage structured effects. What do you do if demography varies by size? What do you do with non-local effects like movement or environmental teleconnections? And what do you do with environmental covariates that affect one another? Kind of merging 3 and 4 here. So the first challenge, the stage structured effects, as an example, I can kind of set this up by pointing out how we do habitat studies for, I think this is POP in the Gulf of Alaska. Here we have a species distribution model mapping juvenile habitat, adult density, and adult growth potential. And so we've got studies that have developed these different maps. But what do you do? What do you do with these 3 maps? They all relate to a single population. And so if you're gonna talk about new shipping impacts, new restoration, it's not just going to affect juveniles in isolation, it's going to affect the whole population. How do you combine across those stages? And as an alternative that has not been pursued but could be, we could define all these different processes at every location and do what's called a Leslie matrix to calculate an intrinsic growth rate at every location and define essential fish habitat from that. Alternatively, we could parameterize an individual-based model, run that to equilibrium and define production from that. Those are both, one of them is simpler than the other, but they're both ways of rolling up all of these different things we know about habitat across stages. For challenge number 2, we and others have made progress on this. This is a results from a paper with Lorenzo Cianelli and Mike Lizzo in 2019, where we fit what's called an empirical orthogonal function, or EOF analysis, to survey data for 14 species in the eastern Bering Sea. And all of those species, all 14, end up with this map of responses to major modes of ecosystem variability. And those modes of variability are shown in the top right. And it turns out that estimated mode of variability just from survey data is extremely highly correlated with cold pool extent. So we know from decades of process research that the production of ice in this ecosystem causes sinking of colder than freezing water, and fishes like cod avoid that freezing water, other fishes seek it out. But all of them are responding to this ice factory. And they don't respond necessarily to exactly the local temperature. They're responding to a regional process, in this case measured by cold pool extent. And they all are responding to this teleconnection. So in this case, we have an example of a documented process research that supports this sort of ecological teleconnection that we could include in our habitat models. Finally, there's this question of what do you do with, this is the third challenge, what do you do with cases where you have really complicated multi-causal processes, so like if age 0 pollock might survive over the winter based on how much they've fed in the preceding summer and fall, and we can measure that directly using RNA DNA ratios, or we could kind of get a proxy based on morphometric condition. We can measure their stomach contents, and we can try to relate that to prey densities, copepods and krill. And doing this, there's a lot of questions. Even after one of the best studied fishery species in the world, we still wonder, are copepods more important than other forage for winter young of year growth? Is RNA DNA ratio a better proxy than morphometric condition for foraging, and so on? And so this, here there are these arrows shown. This is kind of like a quantitative network model for how these different things that we can measure might respond to one another. And we can actually do this in a species distribution model using what's called structural equations. And so given a set of arrows that we hypothesize matter, we can make this effects matrix in the bottom right, where different parameters represent different arrows, the strength of different arrows. And then we can plug that into that equation in the top left. I'm happy to respond by email about this, and it's spelled out in the paper. And we can fit this as a structural equation model. So the point is that a species distribution model can be done to do what structural equation models used to be doing. And we can use them to answer these questions of multi-causality, different cases where we might have multiple mechanisms of, if we change habitat here, we might affect productivity through multiple causal pathways. So, without spending too long on it, the grand challenge for habitat science, as I've been saying, is to predict impacts of localized human activities on population scale productivity. And that matters because then we can weigh trade-offs. We can weigh the trade-off of a new wind farm against, changes in, fishery quota for species in the Northwest Atlantic. We can weigh red tide impacts against recreational fishing, against habitat restoration in the Gulf of Mexico. I think this has always been sort of the holy grail of habitat science. And to do it, we need to step beyond these sort of, pressure indicator response frameworks into this sort of synthesis framework. To do that, I think we combine stock assessment and habitat. And, kind of reiterating, we have to deal with this challenge of stage-structured responses. We can do that by assembling stage-specific habitat responses and combining them. We have non-local responses, and we can identify modes of regional variability, teleconnections, or explicitly model movement. And we've got mechanisms linking environmental variables where we can address that with structural equation modeling. So, with my remaining time, I guess I just want to take a minute to talk about two alternative things that we've been exploring. So the first of them is this this movement model that colleagues and I have published in Fish and Fisheries. It's a high-resolution movement, estimating high-resolution movement rates and habitat utilization from environmental variables, tags, fishery catch and effort, and resource surveys using advection diffusion-taxis modeling. So how do we combine all the information we have about movement to get a single picture of where animals are moving? And so, this is just a really quick kind of brainstorm of, obviously I have an issue with this section of the PowerPoint. But here we have a table showing many different types of data about movement. We have conventional tags, survey data, fishery data. We increasingly have electronic archival tags. We have movement gates like upward facing acoustics or weirs. We have laboratory experiments, selection experiments. We have chemical and genetic and parasite markers. And we have food habit sampling. And all of these give a slice, they give a single picture of movement. And we need to integrate them all to get, best available science as mandated by MSA. And this ends up being really important, not just for habitat, but for stock assessment, for climate research, and for EBFM. So how do we do this? Well, this section is going to be a bit more mathy, but we can decompose an instantaneous movement rate into a diffusion process, a taxis process, and an advection process. And then once we've got an instantaneous movement rate, we can exponentiate that. We can transform it using this operator called a matrix exponential to get a movement fraction, a seasonal or annual proportion of how much they can move from this place to any other place. We can use that to project how abundance will redistribute over time. And there's some kind of math properties about this conserving abundance. It does what we want a movement model to do. And so the key here is that it's defining a habitat preference function, H, as a function of measured environmental conditions. So we then use that habitat preference function, H, to define taxis. And we kind of circularly define taxis as movement towards preferred habitats, and that's shown in math there. The key is that we can estimate a set of parameters representing their preference for different habitat conditions. So habitat preference is explicitly estimated in the model. And so how does this work in detail? Well, in the top left, you might have environmental layers shown in brown or blue, and the resulting environmental preference in black, where they prefer black, they kind of go towards the east or the west, the right or the left on that plot. We can combine that with a diffusion matrix, and that generates a taxis matrix, and we can look at movement for an animal that's released kind of in the bottom row on the kind of left-hand side, the middle, or the right-hand side. And after one time step, they're pretty close to where you released them in the gray bar. That red kind of line is pretty close to where we release them in this gray bar. But over time, they all converge on the stationary distribution. As time goes to infinity, they all go to this black line in the bottom row, because as time goes to infinity, all these animals evenly mix. And so that's sort of a useful property where we could use measurements of movement for a bunch of animals, and we can back out their long-term, utilization of habitat. That stationary distribution is a description of their habitat. We can then fit this in a model with spatially correlated residuals, where we've got a Tweedy distribution for survey data and a gamma for fishery data. And I won't get into too much more of the math, I don't think. Well, okay, I'll skip this about how we fit conventional tags and movement gates, but there's kind of different observation models that are appropriate for different data types. So I think that's all the math I had in here. So to show this in detail, I apply it to the Bering Sea Pacific cod. It's a major fishery, fitting it to the eastern and northern Bering Sea. So the northern Bering Sea used to be ice covered, during the winter, and also to a certain extent in some summers. And that's really no longer the case. Climate change is already affecting, via polar amplification, it's affecting the Bering Sea and the Chukchi and other ecosystems very quickly. And so because of this ice-free Bering Sea, Pacific cod, this major fishery, has moved nearly 1,000 kilometers north over just the last decade. The northern range limit has moved nearly 1,000 kilometers north. So we want to try to understand what's happening and why, what it's responding to. So we fit this movement process that I just described to about 35 years of data in a separate summer and winter season. try to explain habitat preference based on bathymetry and summer or winter bottom temperature from ROMs. And we fit it to survey data, longline fishery CPUE, and conventional tagging data using longline fishery effort as an offset to describe recapture probabilities for conventional tags. So there's an R package called ATM that does this. I'm just showing kind of canned output from the R package. Here is sort of the habitat preference that's estimated in these kind of blue or pink line with a confidence interval showed as shading. The left-hand panel is the preference as a function of bottom temperature, and where they prefer higher habitat preference, they want to go towards the higher number. And so, you know, on the left-hand side, it's saying that their habitat preference really drops off at, you know, it's estimated in this model to drop off at cold temperatures. It might be about kind of steady 0 to 4, and then it might even increase a little bit again after that, although the intervals are wide on the right-hand side. And then in terms of bathymetry, the model predicts that there's a, you know, again, they kind of avoid deeper waters, you know, waters above a kilometer deep. And there's maybe a somewhat more broad depth distribution in winter than summer, but the model is not, with this data, able to sort out too much. So in terms, you know, we can then use this model to conduct experiments about what would happen if we released a fish in different times. The left-hand column is showing a release in the kind of southern middle domain of the Bering Sea in 2017, or the right-hand side in 2014. And the model does not do a great job of distinguishing between movement in a cold stanza on the left and a warm stanza, a warm sequence of years on the right. But it does capture this sort of diffusion of, you know, this predicted diffusion of the animal over time. In terms of preferences, it shows different habitat preference in summer or winter on the left two columns. And then it has the model's also predicting the log of population density, numerical density in the summer or winter. And there is sort of, you know, the top row is 2002, a cold, let's see, yeah, kind of a warm year. 2012 is a cold year, and then 17 and 18 are kind of warm years again. So in the cold year in 2012, the predicted density is shifted kind of southward, whereas by 2017, the density is really getting up there towards the Bering Strait. The model predicts this in both the summer and the winter. We can kind of look at residuals like we would in any model. You know, without going into too much detail, the conventional tag residuals look fine. The bottom trawl residuals and the longline fishery residuals also look generally fine using these sort of newer Dunn-Smith residuals that we're using increasingly. And then the key part, you know, this is sort of the key question for fisheries management, is explaining what proportion we think is in the Eastern Bering Sea. So it used to be that we think this stock was mainly in this Eastern Bering Sea ecosystem. And recently we detected in 2017 that a huge amount had shifted into this formerly ice-free Northern Bering Sea. And so, you know, the top right panel is a model estimate of the fraction in the Eastern Bering Sea that moved to the Northern Bering Sea, that's in blue, or moved from the Northern Bering Sea back to the Eastern Bering Sea. And so, you know, we basically estimate that a very small fraction have used to move from the Eastern Bering Sea to the Northern Bering Sea, and almost all, you know, nearly 50% of them that were in the Northern Bering Sea used to move back to Eastern. And so that would result in a proportion in the Northern Bering Sea that's very low, like maybe 2%. Over time, however, since 2010 in particular, the fish that moved to the Northern Bering Sea are basically kind of happy there. They stop needing to move back. And so that red line drops rapidly after 2010. And that's the model's way of explaining why there's this increasing proportion in the Northern Bering Sea, or a decreasing proportion in the Eastern Bering Sea. And there's different ways of kind of slicing the model to back out what that predicted proportion is. And that's shown on the bottom right. I won't get into too much detail about that, but basically the model is able to capture some of the movement from the northern to the eastern Bering Sea, from the eastern to the northern Bering Sea. We can do kind of a sensitivity of how much the model cares about different data types. So here's sort of a plot of movement over 5 years, summer to summer movement for a fish released in the summer. That's the left column here. And if we exclude fishery data, we get a picture that looks really similar to what we had before. If we drop the survey data, let's see, it also can kind of look similar to what we had before in terms of movement, 5 years of movement. If we drop the tagging data, the model really loses any ability to get at diffusion. And so we need the tags. We need tags for habitat studies because that's the way we can get at how much populations mix, how much fishing here is going to affect population abundance somewhere nearby. So that should be fairly intuitive. That's what the model corroborates. And then if you look at sort of the proportion of the Eastern Bering Sea, on the right-hand column, without fishery data, it looks pretty similar to what we had before. If we drop out the survey data though, the model no longer really captures anything useful about the proportion of the eastern versus the northern Bering Sea. In fact, it gets this wacky picture and thinks that cod have always been in the north, which is not true, we don't think. And then if we drop tags, you know, a diffusion rate slows down, and so certain metrics of movement also change. So, you know, basically what this is saying is that if we have this sort of statistical synthesis, we can figure out what data matter. And in this case, for Bering Sea Pacific cod, the survey matters. This multi-decadal survey matters a lot, and their tagging program matters to understand movement. We can take this and plug it into a stock assessment model. So this is the data is shown in these black dots. This is from a stock assessment, I think from last year. And then the gray is sort of estimates of the proportion in the Eastern Bering Sea, this gray line, in kind of a two-box stock census model. So a stock assessment model that tries to estimate movement. And that model kind of erroneously, again, it can't make sense of the data very well, and so it thinks that there was, you know, 20 to 30% of the population in the Northern Bering Sea, even though there was really no data to support that. The blue line, by contrast, the exact same model, but forcing it to use the movement fractions that this, you know, estimated. And that model also does not do a very good job of capturing recent patterns, but it does do a better job of capturing historical proportions. So, you know, basically, I guess the point here is that we can take output from a movement model and we can plug it directly into, you know, it's not just useful for habitat, but it can be reused also for stock assessment to figure out, you know, to improve a stock assessment model for this commercially important species. So, you know, the point is that we can fit all of these various data types for a synthetic picture of movement. I think that, you know, my kind of cartoon of this is that we don't typically do this. You know, we have a group that will be doing, you know, conventional tagging, and we'll do tagging synthesis. We've got a separate group that might be doing archival tags and does Markov reconstructions of that. You know, and then our assessments in EBFM are typically done with these surveys or fisheries predominantly. We increasingly need to figure out how to do all of these kind of to get a synthetic picture of movement, because climate change is causing rapid distribution shift and is really going to scramble our conventional fisheries management. And then finally, I'll end with a description here of a how we can, you know, sort of a more user-friendly approach to doing stomach analysis. So basically, another part of this grand habitat challenge is what happens with, you know, with predators affecting habitat. You know, as in the North Pacific, as we have large increases in the abundance of some marine mammal predators, how is that going to affect habitat? You know, this is a similar challenge in other areas where some predators are going up, other predators are going down. If we ignore that, we are going to get a very biased picture of habitat quality. And so there's sort of a need for more user-friendly ways of looking at stomach contents or other food habits data. So in this case, this is a project done with Yumi Aramitsu, Tal Levy, and Gretchen Roffler. And it develops an R package called MV-Tweety that I'll explain in detail. So again, this, the theme across this is more kind of synthesis across different types of data and different programs at NOAA. So if we're going to look at food habits, we will need to be looking at stomach contents, for fish and for mammals, and for birds, seabirds. We get a lot of stomach contents from necropsy, on-deck capture and sorting. We get some that's non-lethal, stomach pumping for some protected species, and in other cases we get necropsies of individuals that wash up dead on shore. There's direct behavioral observation, like bill loads of seabirds where they bring prey fish back to feed their chicks. That's not necessarily a direct measure of what the adult is eating, but it's likely to be somewhat informative. And then there's increasingly these kind of geochemistry approaches, so stable isotopes, fatty acid markers, and all of these have different sampling methods where we can do visual identification, genetic, quantitative PCR, metabarcoding, stomach temperature loggers, etc. So there's this huge explosion of new technologies to do food habits work, and we again need a way to do synthesis of it to understand how it affects habitat impacts. So methods to analyze food habits data, there's been a lot of discussion over this, of course, in ecology forever. The simplest way is just to take an average of the encounters, like the % frequency of encounter in stomachs, or take the proportion of biomass in stomach samples or scat. And this is a problem because that spatially imbalanced sampling will cause a bias. We don't go out and randomly sample scat. We sample scat where we can encounter it. And so we need to back out sort of the bias caused from our sampling process. There's a hierarchical expansion from post-stratified samples using predator densities, but it can, so it's basically taking what we always have done and doing it by strata and then adding those up. That's like kind of what the Alaska Fisheries Science Center has been doing for food habits for a long time. However, it ignores fine-scale heterogeneity or continuous predictors. It doesn't work like a model, and so we can't bring in a lot of the kind of modeling insight we might have. Recently, there was proposed a different class of models called a Dirichlet model. It allows sort of all these regression techniques, but it requires modifying the data before you analyze it, and then kind of hard to interpret parameters. And in this paper, we're introducing a different approach that's just a generalized linear or generalized additive model using a Tweety distribution, as I'll try to explain. And it basically allows all our sort of standard regression analysis approaches. So developing this requires kind of a short tangent into point processes. So if you've got, and this is basically sort of a way to represent animal foraging. So let's imagine that there's a predator that's going across a field of prey. There's prey distributed spatially, and the location of each prey is random. They're moving around. If we make that slightly more specific as a Poisson process, that means that every prey is moving independently and randomly, so that they follow a Poisson distribution, and we can put covariates in that. We then make this slightly more specific as a marked point process. Every prey animal has a size, follows a gamma distribution, and follows some prey species that we also can measure. And let's say we only sample some of these points. We can only see some of the prey that are captured. Capture probability may vary among prey. And that results in a thinning rate that's basically like a kind of a nuisance process. This is all what I'm calling a thinned and marked point process model. And we can approximate that really conveniently with what's called a Tweedy, or kind of more specifically this like multivariate Tweedy distribution. So if you've got a point process, I'll walk through this in detail. So if we've got 3 prey that are distributed across space, in the top row we've got prey 1, 2, and 3. Prey 1 might be centered in kind of the middle. So these kind of circles are showing areas of highest density and then lower density as you move away. And the dots in that kind of purple color, or kind of, yeah, maybe purple color, are the location of prey and their size. Prey 2 is in green and kind of is centered at this habitat patch on the top left. And prey 3 in yellow is kind of centered on this kind of middle left. Again, showing the size and distribution. And let's say we've got 3 different central place foragers. One of them is in the kind of top left, one's in the middle in green, and one's in the bottom right in kind of blue. So if we look at a bunch of realizations of sampling for those central place foragers of those different species, and then we get this, we can simulate from that, and we get these distributions shown in the second row, and a single sample is shown in these kind of vertical lines. We can then approximate that true distribution from this thinned and double marked porcelain point process as a Tweedy distribution, and I won't get into the parameter matching, but these lines are the analytic calculations from a Tweedy distribution. And so, like, for instance, the third row, the yellow line and yellow dot looks pretty close to the yellow histogram and yellow dot of the second row, and so on. So we can take this kind of detailed foraging process and approximate it as a Tweedy. And then if we fit it as a Tweedy glim, that's shown in the bottom row. It's sort of, there's some restrictions on parameters that the Tweedy distribution involves to fit as a glim. And so it changes the proportion of zeros a bit, but you still get a close representation of foraging. Yeah. So why care? The point is that we can think about this kind of idealized process of a predator foraging on prey animals of different size, and we can fit that as a glim or a gam, and then back calculate the proportions of different prey using kind of standard off-the-shelf software and visualization tools. So it's a lot of kind of complicated sounding math, but in the end, it just gets us to the kind of stuff that anybody could run on their machine if they want to. So for instance, this is, it's truncated on the bottom very slightly. There's an extra row on the bottom that's not shown, but in this case, there's a new R package, MV Tweedy, where you can just load data for this Middleton Island puffin example that I'll show. We can fit a GAM using MGCV, the Tweedy family. We can tell it what the class is. This class is MV Tweedy. We can predict things, and then we can plot it using ggplot. And so if we do this for tufted puffins on Middleton Island, including sea surface temperature as a covariate, we can predict what is the proportion of diet of these different prey species given the local temperature. And the model is basically saying that in higher temperatures are associated with increased proportions of Pacific herring, significantly increased herring, and lower temperatures are associated with significantly increased walleye pollock or prowfish. And looking in the top right at the proportions, we can see that in the 80s, 1980 through 2000, there was sort of this oscillation from Pacific sand lance and prowfish that puffins were eating. Whereas later, there's this sort of oscillation between Pacific herring and capelin. And yeah, so we can kind of have this model to reconstruct food habits of puffins in Middleton Island. As a contrasting example that's maybe more relevant to habitat, we can do the same thing using wolf scat in Southeast Alaska. This is what Tal Levy and Gretchen have been doing for a long time, where they sample scat and then they use what is called eDNA metabarcoding. And here we're compressing that down to 10 major taxa and plotting it with ggplot. And, you know, basically the story is that ungulates, these black-tailed deer and moose in the top row, are important prey throughout the distribution. But the kind of fun piece is that marine mammals, there's sort of islands that are, I should have used the updated plot here that grays out some of these islands that are not actually colonized, but there's islands where marine mammals are a large part of the diet of wolves. There's other islands where beavers in the kind of eastern and southeastern part of Southeast Alaska, beavers are a large part. And then there's kind of headwaters where fish make up a large part of wolf diet. So this is all to say that, you know, we can use stomach contents and use these kind of new synthesis methods to get a map, a predation landscape, and use that to understand how, you know, in this case, you know, hypothetically reintroduction of wolves or changes in wolf hunting could affect population productivity for these different prey species. So, you know, in conclusions to take away from this, fitting food habits can be as simple as a GAM or GLIM. You know, we typically have a team at every science center that does food habits work. And in my experience, it's, you know, intimidating to try to unbox those data, not being like a specialist. And so this is sort of a way to try to make it easier for people to unbox stomach content analysis. We can include covariates that affect density. Some of those are sampling covariates, some of them are habitat quality. And we can think critically about what things we're including. And it's easy to estimate kind of spatial or temporal variation in food habits using these types of models. So we can do regression methods, or ultimately it'd be great to combine it with the movement model and this kind of middle section that was already discussed. So without further ado, I guess I'll end with just a single slide about what I think this says for NOAA. And it's, again, slightly cropped on the left, sorry about that. So I guess my claim is that, you know, NOAA, you know, based on this, we know that, I mean, all of these examples draw upon long-term surveys and long-term habitat studies. You know, all of us, again, are doing this relay. Science is a relay where we do methods development, technology development. We maintain surveys that are longer than the lifetime of a scientist. And all we can do is hope that we, you know, pass the baton. But these, you know, these surveys are really the crown jewels of NOAA, you know, and we need to find ways of supporting them, not just for habitat, not just for ecosystem, not just for stock assessment or climate analysis, but for all of them. Specifically for habitat science, I think that this suggests that there's sort of a need for habitat science implementation plan. We've done an implementation plan for, you know, as I understand it, for our climate work. We have a climate strategy, and then we have a, you know, separate kind of regional implementation plans. We have separate policy documents explaining how to do that. You know, habitat science, by contrast, has had sort of a habitat research plan, and there, I think there's a need for us to develop sort of best practices for how to achieve the goals in that habitat research plan. And then I think that there's sort of, you know, given this huge increase in data, you know, in technologies for sampling movement, for sampling food, we're going to need to support development of these kind of unified analytical tools that can apply across regions. You know, no single regional science center is going to be able to do all of this on their own. And so we're going to need to, I think, coordinate across science centers to develop new techniques for habitat synthesis. So with that, I'll put up my email at the top. There's a lot of collaborators. These, you know, if I listed all the people who contributed data, this would be even longer, you know, in terms of surveys. But thanks to people listed here. Thanks again to Peg, and I think I have a few minutes for questions.
Yep, thank you for presenting, and we have a couple of questions here. I just want to remind the attendees that you can type any questions you have in the question panel, and we'll get to as many as we can. So the first one here says, thank you for throwing out this challenge. Your region has a wealth of data that other regions may not have. Any suggestions regarding approaches that would be appropriate for data poor regions?
Yeah, thanks for asking. My feeling is that there's sort of a data spectrum in terms of, different regions have different data more or less. And then different regions have different kind of habitat needs, they need different information. And, so again, there's sort of this massive wind farm buildup. There's sort of estuary restoration in the Gulf of Mexico. The California current has its own set of challenges, and then Alaska and the Pacific Islands, each of these has different needs. And so both the data differences and the different science needs means we're always going to have to look at regional differences. But to generalize a bit, I do think that just like in stock assessment or an EBFM, we've come up with this data poor to data moderate to data rich strategy. We could do the same for habitat. In some cases, it could be as simple as taking, life history theory and meta-analyses to look at sort of expected habitat response based on taxonomy and based on similarity of, and FishBase has some habitat information for every, could be used to predict habitat for every described fish. That would be the most data poor method, but even that would be somewhat more analytically fleshed out, I think, than the way we've done, the habitat vulnerability analyses. If we do that by stage, we could also start doing that sort of like Leslie matrix approach to integrate across stages. And so that, that sort of data poor method, whatever it looks like, I think could be done for all species in a, a given fisheries management plan as mandated by MSA for EFH, essential fish habitat work. But in our region, we're discussing doing these kind of Cadillac habitat assessments, key species, where spatial management of Bristol Bay red king crab or other cases where there's a really pressing, habitat question where we can spend the time to do more than that sort of data poor method. And that's where we'd really get into some of the more analytically complicated stuff that I've been discussing, I think. Anyway, I'd be happy to chat more offline with whoever asked.
Yeah, and I will forward these questions and any questions we didn't get to you at the end of the presentation, so you can follow up with people if you'd like. The next question, if and when funding is limited and we cannot support every type of data collection or analysis, what would you suggest as the top activities to focus on?
Yeah, I mean, of course, like how to allocate things is the hardest challenge that our agency probably has, I think, because, our greatest asset besides our surveys is our staff, and, making these decisions in a fair, transparent, and equitable way, I think is incredibly challenging. At the Alaska Center, we've been kind of trying to make these sort of quantitative kind of cross-program analyses that can really guide what is the key data for a given problem. So, we've done that in the survey team to figure out if we can get away with dropping a third boat in the Gulf of Alaska. In other cases, we've, developed new adaptive surveys for our, spring ichthyoplankton sampling. I guess the bottom line is that we're trying to improve transparency by actually developing a pipeline where we can prioritize things, not just people in a room saying, hey, this is more important than that, but actually coming up with sort of an agreed upon and documented pipeline for what's useful, what's most useful and what's kind of great to have, but not quite as central.
All
It's
right,
kind of a
so
dodge.
we are coming up close on time here, so I am going to forward any questions we didn't get to. I will forward those to you so you can follow up. And I just want to say thank you to our speaker today, and thank you all for joining us. You'll be able to find this presentation on YouTube later this afternoon. When the presentation ends, you'll see a survey. If you take time to fill this out, we do use these surveys to improve marketing our library seminar service and inform future seminar topics. Thank you all again for joining us, and have a safe and enjoyable rest of your day.
Yeah, thank you.
Thank you, Jim. Excellent presentation. Thank you.